Hey everyone, so in this video I'm going to discuss about another DNA manipulative enzyme named DNA polymerases. As we all know that DNA polymerase may copy, add labels and fix the DNA, and here is E. coli DNA polymerase 1, the classic DNA polymerase. It moderately processes polymerase and it has an activity 3' to 5' proofreading, exonuclease activity and 5' to 3' strand displacing and nick translating exonuclease activity, and it is used mostly for labeling DNA molecules by nick translation, and for other purposes the clinal fragment is usually preferred. Here the enzyme named DNA polymerase 1 has, in addition to its polymerase function, it has 5' to 3' and 3 to 5' exonuclease activity. A major use in the enzyme is in the nick translation procedure for radio labeling DNA. The 5' to 3' exonuclease function of DNA polymerase 1 can be removed by cleaving the enzyme to produce what is known as the clinal fragment, and this retains the polymerase and 3' to 5' exonuclease activity, but it lacks 5' to 3' exonuclease activity. Now, clinal fragment, the C-terminal 70% of E. coli DNA polymerase 1 discovered by clinal, and it lacks 5' to 3' exonuclease activity and uses into in the radio labeling and labeling DNA termini by filling in the cohesive ends generated by certain restriction enzymes, and it generates blunt ends, and it mostly used in the DNA sequencing. Now, if we treat the DNA polymerase 1 holoenzyme with the protease named subtilisin, and it thrown into proteolysis, it lacks 5' to 3' exonuclease activity. It cuts 5' to 3' exonuclease domain, but it will retain the polymerase domain. It will retain the 3' to 5' exonuclease domain activity, and now the fragment is known as clinal fragment, which only retains polymerase activity and 3' to 5' activity, but it lacks 5' to 3' activity, so the fragment is known as clinal fragment. Other one is reverse transcriptase, and RDase is an RNA dependent DNA polymerase, and therefore produces a DNA strand from an RNA template, and it has no associated exonuclease activity. The enzyme is used mainly for copying messenger RNA molecules in the preparation of complementary DNA, cDNA or copy DNA for cloning, although it was also act on DNA templates. And reverse transcriptase is a key enzyme in the generation of complementary DNA from RNA template, and the enzyme is an RNA dependent DNA polymerase which produces a DNA copy of an mRNA molecule. There are also enzymes that modify the ends of DNA molecules. We call them the modifying enzyme. The enzymes named alkaline phosphatase and the polynucleotide kinase, that is T4 polynucleotide kinase, and terminal transferase act on the terminal DNA molecules and provide important functions that used in a variety of ways. The phosphate and kinases. The phosphatase and kinases enzymes, as their name suggests, are involved in the removal or addition of phosphatase group respectively, and bacterial alkaline phosphatase, there is also a similar enzyme called intestinal alkaline phosphatase, removes phosphate groups from the 5' end of DNA. Here you can see modifying enzymes. Number one is alkaline phosphatase. It removes a phosphate group from 5' and the triple and here it removes and it replaces it with hydroxyl group. Next is polynucleotide kinase. It also removes phosphate group and replace it with phosphate group. It removes hydroxyl groups and replaces it with phosphate group. And there is a terminal deoxynucleotide transferase which add nucleotide sequence in the 3' end and we say in the hydroxyl end. Next is host control restriction and modification. Restriction systems also bacteria to monitor the origin of incoming DNA and to destroy it, and if it is recognized as foreign, restriction endonuclease recognize specific sequences in the incoming DNA and freeze the DNA into fragments, either at specific sites or more randomly, thus preventing it from successfully replicating and parasiting the cell. Here is a restriction and modification enzyme. Restriction enzymes widespread among prokaryotes and rare in eukaryotes and protect prokaryotes from hostile foreign DNA. Example includes viral genomes and essential for in vitro DNA manipulation. Restriction and modification enzymes include three classes of restriction enzyme. Here is type 2 cleaved DNA within their recognition sequence and are most useful for specific DNA manipulation. Restriction enzymes recognize inverted repeat sequences, that is palindromic sequences, and typically 4 to 5 base pairs long. EcoR1 recognize of 6 base pair sequence we will discuss in the previous lecture and generated sticky ends and blunt ends. Restriction enzymes generate one of the three types of end, that is 5' overhang, and there is 3' overhang and blunt ends. In the previous video, I had discussed the 5' overhang, and it occurs like in this manner and generate 5' overhang and 5' extension. 3' overhang, it cuts the DNA nucleotide sequence in this manner and generates 3' extension. And next is blunt ends, that cuts the double stranded DNA from in equal position and equal size. No generation. It doesn't generate 5' to 3' end, and the enzyme that perform the cleavage is SMA1. Here is the generation of blunt and sticky ends. These are the examples of recognition sequence for restriction enzymes. There are 4 cutters, 6 cutters, 8 cutters, and what type of end they produce are blunt ends, 5' overhangs, blunt ends, 3' overhangs, 5' overhangs, and these are the enzymes that produce these types of ends. And in the last, here is a chart that shows the cutting and modifying and joining DNA. It requires enzymes which are catalytic proteins molecules and purified from wide range of cell types and are available from commercial suppliers. Enzymes are divided into 3 main groups, that are restriction endonuclease, DNA modifying enzymes, and DNA ligases. And restriction endonuclease that cut DNA at defined recognition sites, and modifying DNA enzymes such as nucleases, polymerases, and enzymes that code at which joins DNA molecules via phosphodiester linkage or to generate recombinant DNA. Let's discuss the restriction endonucleases that code DNA at defined recognition sites and are used for generating DNA fragments, and restriction mapping that we will discuss in our next videos. And next is DNA modifying enzymes such as nucleases. Nucleases are divided into two types, an endonuclease and exonuclease, such as restriction enzyme DNase 1 and S3 nuclease, and exonuclease such as BAL31 and EXO3, such as DNA polymerase and clean-out fragments, like fragments of 3' exonuclease activity that I have discussed, and reverse transcriptase that generates cDNA from RNA molecule, and third is enzyme that act on termini such as alkaline phosphatase, polynucleotide kinase, and terminal transferase. Alkaline phosphatase removes phosphate groups, and polynucleotide kinase adds phosphate groups, and terminal transferase add nucleotide sequence at 3' ends and hydroxyl ends. So in this video I had covered two more manipulative enzymes, and if you like this video, hit the subscribe button and like the channel for more informative